two-component systems have. First, sensor protein called sensor kinase is located at the outer membrane or cytoplasmic membrane and it monitors environmental parameters. The sensor protein has N-terminus called the input domain and C-terminus called the transmitter domain. The transmitter domain of sensor kinase has a conserved histidine residue attached to and is capable of autokinase activity, attaching phosphate group from ATP to histidine residue. The phosphorylated histidine then can transfer the phosphate group to aspartate residue attached to the response regulator, which is the second protein of our two-component system. The phosphorylation of response regulator causes it to undergo a conformation change that serves to elicit response. Typically, the response regulator binds to DNA and modulates its transcription. Now, we are going to talk about the regulation of porrin synthesis in E. coli. Porrin synthesis is influenced by 1. pH 2. Osmolarity 3. Temperatures 4. Toxins 5. Growth phase. However, porrin in E. coli comes in two different diameter. First, when MPF is expressed the porrin diameter is about 1.2 mm, while when MPC is expressed the porrin diameter is about 1.1 mm. When the cell is in an environment where there is a higher level of taxon such as the human intestine, the cell undergoes a change and expresses MPC for smaller porrin diameter. But when the cell is in an environment where there is a lower level of taxon such as fresh water, the cell expresses MPF for wider porrin diameter. Now, going back to our two-component signal transduction, when it comes to porrin regulation in E. coli, the sensor kinase is called NVZ while the response regulator is called the MPAR. Thus, one can say that the MPAR can be phosphorylated and unphosphorylated. When there is a high osmolarity outside the cell, the level of phosphorylated MPAR increases, while when there is a low osmolarity outside the cell, the level of phosphorylated MPAR decreases, and this has a significant impact on what size of porrin diameter will be expressed. When there is a high osmolarity, MPAR binds to low affinity regions upstream of MPC promoter, thus activating the transcription of MPC. On the other hand, MPAR also binds to low affinity and high affinity regions upstream of MPF promoter, resulting in formation of protein-protein repressive loop preventing the transcription of MPF. However, when there is a low osmolarity outside the cell, the level of phosphorylated MPAR is low, therefore, MPAR binds to high affinity regions upstream of MPF promoter and activating its transcription, while MPAR does not bind to upstream of MPC because there are no high affinity regions upstream of MPC.
Now, we are going to talk about another way of regulating porrin synthesis. This time it's about how temperature affects porrin regulation. Introducing MCF, a post-transcription negative regulator of MPF. It is an antisense RNA that complementarily binds to MPF, therefore, the ribosomal binding site is blocked by MCF, preventing the translation of MPF. It also decreases the stability of MPF mRNA. Also, the transcription of MCF increases 10 times higher when the temperature is 37 degrees or higher. So what activates the transcription of MCF? First, SOX-S, which is part of the SOX-RS regulon. SOX-R is activated by oxidative stress, and when it is activated, it now can activate the transcription of SOX-S. Then SOX-S can now bind to MCF promoter and activate the transcription of MCF. Second, MAR-A, which is part of MAR-RAB-operon that is controlled by MAR-R. However, in the presence of salicylic acid and antibiotics, MAR-R is repressed. Therefore, MAR-A is allowed to be transcribed and it then can activate the transcription of MCF. Lastly, RAB, which is a putative transcription cofactor. It is believed that it is activated by cationic peptide antibiotic by unknown mechanism. A post-transcription regulator of MCF is a protein called STPA, which is a chaperone protein. It controls the stability of MCF RNA by binding to it and altering its conformation to be more susceptible to nuclease degradation. Another factor that affects the level of MCF in the cell is whether the cell is in nutrient-rich media or in nutrient-poor media. When the cell is in rich media, the level of MCF are high, therefore, MPF levels are low and vice versa. A protein called LRP or leucine receptor protein plays a major role in repressing MCF transcription in nutrient-poor media. Overall, when the cell is in rich media, meaning the nutrients are high, so STPA levels are low, therefore, low levels of repressor of MCF resulting in increasing numbers of MCF repressing the MPF giving the cell to express MC instead. This also occurs in vice versa, and that's where the video ends. Good luck to everyone. Happy studying.